All right, I guess we should start. Really super excited to have John here today. It's been Mark and I have been planning this for a long time, and this is probably our best February, right? We have John starting out February, and then we have in the February 27th, we have Eric Betzig talk as well. So it's really a phenomenal month for us. John grew up in Northern Virginia. So if you don't know, Janelia is in Northern Virginia. And he went to the University of Virginia to get his BS in chemistry, and then went to grad school in the University of Wisconsin in Madison, also in chemistry. And then he moved to Boston to be a medicinal chemist at Merck Research Laboratories. And in 2010, he came to Janelia to apply all his medicinal wizardry to a really old field of dye chemistry, century-old field, and is really the co-founder of the GF dyes. And we're super excited to have him talk today. John?
All right. So I'm glad that I'm going before Eric. So before the Nobel laureate. But yeah, so I'm John. I've been a member of Luke's lab for almost 14 years now. I started in 2010. I want to thank Mark and Brian for the invitation to speak as part of this seminar series. And so what I'm going to do today is talk to you about almost really the entirety of a program that we've been working on in group for really over 10 years now. But before I dive right into that, I think it's important to talk about what we are. Now, the Janelians in the audience, I'm sure know this. They've seen Luke talk about this many times. So I apologize if you're seeing this for the 10th time. But we are a group of synthetic organic chemists. And the way that we fit in at Janelia is as tool builders. So we use synthetic organic chemistry to build the types of tools, predominantly fluorescent dyes and probes based on those, to enable the types of sophisticated imaging experiments, biological studies that our collaborators, coworkers, and end users are interested in doing. So much of our time is spent really on the chemistry portion here in terms of optimizing old chemistry and often coming up with new chemistry to access or better access the types of tools that we want to build. And naturally, the other half of this is that as we build these probes, these dyes, we have our end users try them, they give us feedback, and then we go ahead and optimize and iterate upon those tools to best facilitate the types of studies that they want to do. Okay, so this is not an audience, I think, where I need to belabor the point about what fluorescence is, but I just kind of like to include this as a sort of warm-up to the talk. I think we all know that a fluorophore is a small molecule or chromophore that absorbs a photon of light, moves into an excited state. It can relax back down to the ground state through the emission of a photon of slightly longer wavelength light. Now, when we're talking about the small molecule chromophore pi systems here, we're of course talking about visible light. And the way that we typically characterize fluorophores to compare them is first by taking their spectra, their absorbance, and their fluorescence, excitation, and emission spectra. And then we can easily categorize them based on their absorbance maximum and emission maximum wavelengths. So it's handy to have a way to compare the different brightnesses of different fluorophores because they do vary quite a bit in terms of their brightness. And one of the key variables that or parameters that we use is quantum yield. And quantum yield is really just simply the photons that are emitted by a fluorophore divided by the photons absorbed. And so it's essentially just a representation of the efficiency of fluorescence, the maximum value being 1. And for the vast majority of purposes, we want the highest values that we can get. One other variable parameter that we would measure is the extinction coefficient, or the molar absorptivity, which just kind of represents how much or how well a molecule absorbs. And again, if you want bright fluorophores, you want ones that typically have high extinction coefficients. All right, so fluorescent dyes have been around for quite a while now, although fluorescence itself only became more common in the last 70 or 80 years. But these dyes have been around for a long time. But as fluorescence microscopy has advanced and become more sophisticated, the demands placed on dyes have increased substantially. So when you start talking about techniques like single molecule tracking, super resolution microscopy, and even just trying to do multicolor, multiplex fluorescence, it is important to be able to tune, design, rationally plan out dyes that can fit and best support these types of microscopy. So what do we want from our fluorophores? Now, most of these are pretty obvious, but of course, yes, we want typically the brightest, most photostable fluorophores that we can make, because the more photons you get out of a fluorophore before it bleaches or falls apart, the better resolution and the better localization and super res that you can get. We like to have as many colors as possible across the visible spectrum to facilitate multicolor imaging and make it easier, and to get away, in some cases, from some of the UV fluorophores or those far violet blue ones that can be more damaging to living cells and tissues. When we start talking about doing live cell imaging, a key issue is cell permeability. Not all small molecule fluorophores are cell permeable, but this is very much key when you're talking about doing single molecule tracking or doing super resolution in live cells. And lastly, especially when you're talking about small molecule fluorophores, you need a way to specifically label the cellular components that you want to visualize, that you want to image. So we need ways to target our dyes to the structures of interest. So we are organic chemists. We like small molecules. Obviously, fluorescent proteins have had an enormous impact on the field in the last 30 to 40 years. And they certainly have extraordinary advantages. But small molecule fluorophores definitely have their place, particularly because they tend to be amongst the brightest and most photostable fluorophores. And there are a variety of different structures across the visible range and of varying brightness that we can choose from. But one of the most popular scaffolds, and one of the ones that is most interesting to us, is the xanthine scaffold, the xanthine series of dyes. And these are, as you can see here on this scale, very bright, and they encompass, a decent range of wavelengths and colors. So these xanthine dyes include the fluoresceins and the rhodamines, where the main distinction here is between whether these substituents are oxygen on the 3 and 6 positions of the xanthine chromophore, or they are nitrogen. And the reason we like these is, as I mentioned before, they're bright, they're photostable, but as I'll talk about later, they're also highly tunable through relatively modest, small structural changes. And even if you're not talking about fluorescence microscopy, xanthines have been widely used for a variety of purposes in general life. If you've ever been to an ophthalmologist or an optometrist and they've put a yellow-green dye in your eye, it was probably fluorescein, and they do that often to check for damage to the cornea, to check for corneal abrasions and things like that. And rhodamines have been used for environmental and industrial applications, even just as tracker dyes because of their very bright color. Now, one of the properties of xanthine dyes that makes them so interesting to us is that they have this unique equilibrium between a closed lactone form that is non-fluorescent and this open zwitterionic fluorescent form. So it has this charge separation here and this uninterrupted pi system that makes it a chromophore and a fluorophore. So we can characterize this equilibrium through what we call the KLZ, the equilibrium constant of this equilibrium. And I'm going to talk about this quite a bit later in the talk. The main takeaway here that you can just remember is that very low KLZ values indicate a dye that favors the lactone form, whereas higher values favor the zwitterionic form. Now, this equilibrium has a number of consequences for the behavior of rhodamines, particularly in biological systems. One of those is that it imbues them with a type of sort of dynamic amphipathic nature, where these dyes can often be very open in aqueous solution, where the charge separation is more stabilized. They can then close up into the more lipophilic lactone form to slip through plasma membranes, and then open up on the other side to regenerate their fluorescent form. We can also leverage this equilibrium to make fluorogenic dyes. One of those ways is through caging these dyes with groups that lock the dye into the closed form and then can be removed through light or an enzyme or other stimulus. Or you can simply take advantage of dyes that have an inherently low equilibrium constant that will bind to, say, a cognate protein and then open up because of the change in the environment. So what actually surprised me when I first started working on this was just how old these dyes are and the fact that they are still used so readily. So fluorescein was first discovered in 1871 by Adolf von Baer. And the rhodamines were first reported or synthesized only a few years later by a Swiss chemist, Maurice Aersol. What is perhaps even more surprising is that these dyes have predominantly been made through the exact same synthetic route for much of the last 150 years. So for rhodamines, this route typically involves the condensation of an aminophenol with a phthalic anhydride in the presence of a very strong acid at extremely high temperature. By contrast, fluorescein syntheses are a little more mild. Here we have a resorcinol that's condensed with a phthalic anhydride again, but usually we use a milder acid at a lower temperature. Now, one of the consequences of having these extreme conditions here is that this, this synthetic route is not very tolerant of functionality on the aminophenol in particular. So the types of nitrogen substituents that you can introduce into your rhodamine is severely limited by the severity of these conditions in this route. So when you actually do these syntheses, you'll find that generally the fluorescein condensation works pretty well. The yields are high. Isolation and purification of the fluoresceins is pretty easy. When you do it with rhodamines, this is not the case. Often the yields are very low. You get mixtures of products that are nasty, sometimes intractable. And again, those harsh conditions really limit what you can do in terms of functionality. So one of the very first things I worked on when I joined Luke's group back in 2010, 2011, was to consider whether fluoresceins, which are much easier to make, could be converted into rhodamines and entirely avoid these harsh, high temperature, strong acid conditions. And so what we did here was to leverage some modern chemistry and drag dye chemistry kicking and screaming into at least the 20th century, if not the 21st. And so here what we did was to use palladium cross-coupling chemistry. So we could take a fluorescein, we could form a ditriflate where these sulfur functionalities cap the oxygens. Now, triflates are very well established to undergo cross-coupling reactions catalyzed by palladium with a variety of different coupling partners, including amines, to replace the entire triflate moiety with the nucleophile, in this case, the amine. This particular reaction is known as the Buchwald-Hartwig cross-coupling, which is named for the two groups that independently reported it in the 1990s, from Steve Buchwald and John Hartwig. And just to emphasize how important this type of reaction has been to organic chemistry, the 2010 Nobel Prize was awarded for palladium-catalyzed cross-couplings to 3 of the earliest investigators that explored this type of reactivity. So what we are doing here is attaching the entire nitrogen group, which we will often call the oxychrome. We'll use that term a bit more later in the talk, and form this carbon-nitrogen bond at the very last step. And what that allows us to do is incorporate a greater range of functionality and make these in a very divergent way from a single intermediate. And so that's exactly what we did. In 2011, we reported a simple paper where we just couple on a whole variety of different nitrogen nucleophiles, including cages and other enzyme labile groups. And I will return to this chemistry in a minute because it plays a key role in the development of the entire Janelia Fluor dye series. So now I'm going to take a little bit of switch gears a little bit and talk about why we even started working on the JF dyes in the first place. So I think many people in the audience will know that single molecule tracking is an important technique in fluorescence microscopy. It's important for studying molecular dynamics in cells and seeing how proteins move around inside cells while they're actually alive. But one of the important considerations when you're doing single molecule tracking is how you label your protein of interest. And one of the techniques that has become extremely popular in the last 20 years or so is the use of the self-labeling tag enzymes like HaloTag and SnapTag. So for those who are not familiar, the way this works is that you express a construct consisting of the protein that you want to study attached to the self-labeling tag enzyme. You can then administer your fluorophore with a reactive ligand on it that is specific for the self-labeling tag enzyme. And so when these two meet inside the cell, they undergo an irreversible covalent reaction that attaches your fluorophore through, in this case, HaloTag, to the protein of interest. And any excess ligand fluorophore can typically be washed away. And so, yeah, here we have a more specific example. We have HaloTag or chromophore. We watch a protein zip around a cell. Isn't that great? OK. So back in around 2012, we were actually approached by and discussing issues with single molecule tracking with some of our collaborators, particularly some members of Bob Tejan's lab. And they were expressing to us that many of the existing dyes for this have quite suboptimal properties. And one of the most popular dyes used at the time for this was tetramethylrhodamine, so one of the more simpler and older rhodamines around. One of the advantages of TMR is that it is cell permeable, but its brightness is actually quite subpar, especially for a rhodamine, with a quantum yield of 41%. Now, there have been many campaigns over the last 20 or 30 years to improve the brightness, photostability, and in particular solubility of rhodamines, but these have often come at the cost where you have to essentially forfeit cell permeability, because the way this is often done is through the installation of very hydrophilic polar groups like sulfonates. So things like the Alexafluor dyes are not particularly useful for live cell self-labeling tag enzyme technologies. And so this encouraged a question about whether we could use organic chemistry to fix this with TMR, retain its cell permeability, and improve its brightness. So how do you do this? Well, the first thing you have to consider is, why is the quantum yield of TMR low? So TMR is curious when you compare it to two of the other most simple rhodamines, rhodamine 110 and dimethyl rhodamine, which have very high quantum yields, but they only differ in the presence of these additional alkyl groups on TMR. So if you consider the absorbance and fluorescence of TMR, you would expect in the ideal situation, it absorbs a photon, comes back down, emits a photon, it's perfect, we love it, quantum yield 1. Except that's not what happens, because there are non-radiative decay processes at play, and one of the principal ones that we believe impacts rhodamines like TMR is what we call twisted internal charge transfer. And what this process involves is rotation around the carbon-nitrogen bond here, concomitant with the donation of an electron from the nitrogen to the pi system of the chromophore. And through this rotation, it can relax back down to the ground state without emitting a photon. There are other things that can happen, of course, intersystem crossing to the triplet state, which can result in singlet oxygen generation, which can do all kinds of gnarly things to the rhodamines and contributes to their photo-bluing and photo-bleaching dealkylation. I won't really get into that today. That's something for another time. But if we focus on TICT, which we suspected was a major contributor to the quantum yield issues with TMR, we can look a little more closely at what structural and electronic parameters might be predicting, affecting, or playing into driving TICT. So if you look at the length of the carbon-nitrogen bond here, a longer bond is going to indicate less double bond character and indicate a bond that can perhaps rotate more freely. A nitrogen that has a lower ionization potential is going to be more eager to give up an electron as part of the TICT mechanism. And as these hydrogens get closer together, you have, well, for the organic chemists in the audience, a sort of pseudo-allylic type strain, where this is going to want to push these alkyl groups out of plane with the rest of the xanthine system. And so back in 2012 or so, Luke went in his office and did some molecular modeling and found something interesting. The molecular modeling suggested that if we were to tie the nitrogen groups into rings, that this could potentially improve brightness, and that was manifested through the calculated values of the parameters we just talked about. So if you consider the simplest set of azacyclic rhodamines from the 3-membered ring through the 7-membered ring, what we found was that one analog stood out. This is the 4-membered ring or azetidine ring containing rhodamine. And it was predicted to be the best because it had the best values for all 3 of these parameters, including the shortest carbon-nitrogen bond distance, the largest distance between the protons, and the highest ionization potential. But of course, we had to make this dye in order to validate that. And we were faced with a problem initially, which is, well, azetidine is not at all stable to acid, particularly strong acid. If you've taken organic chemistry, you know 4-membered rings like this are not super stable. They are strained, they tend to pucker, and they are prone to opening by nucleophiles or even just by acid. And so this is where the cross-coupling comes back into play. So we were able to take this modern, common now chemistry technique or strategy to now go ahead and make this bis-azetidinyl rhodamine through a really straightforward cross-coupling from a fluorescein ditriflate. And it works really well, the yield's very high, and we were able to use this to make actually all of these azacyclic rhodamines here. And when we measured the quantum yields of most of them, they were actually worse in most cases than TMR. The pyrrolidine was better, but what about the azetidine? Well, there's a reason that I'm here even talking about this, and that is that when we made the bis-azetidinyl rhodamine and measured its spectral properties, we found that this very simple change in the structure resulted in a substantial increase in quantum yield and in the extinction coefficient. And so just by, well, yeah, we called this dye Janelia Fluor 549 And all the dyes that I name in this talk, we name them based on the absorbance max. So 549 is the absorbance max for this dye. So what we've done here is we've just added 2 carbon atoms, and we've improved the brightness of the dye by 3-fold. So it's a pretty atom economical process. When we compared the spectra of these 2 dyes, they are essentially superimposable, JF549 just being brighter. And so any place where you would use TMR, you could slot in JF549 from the standpoint of spectra and wavelengths. JF549 also exhibited superior 2-photon performance and improved brightness. And if we prepared the halo tag ligand of JF549, we could use that to specifically label in live cells, in this case, histone H2B attached to halo tag. It worked very well. And when we compared JF549 and TMR in DStorm, we saw higher resolution and better performance from JF549. Now, James Liu's group looked at single particle tracking and found that JF549 actually gave substantially improved performance with twice the photons for twice as long. And in these movies here, you can play the movies. There we go. You can see here we have JF549 through halo tag on Huntington 25Q, and here on a histone in a live HeLa cell. So this, though, encouraged another question, because there are many fluorophore scaffolds out there that contain these sort of dialkyl amino groups. And so we wondered, well, could we use the same strategy to improve their brightness as well? Not to belabor the point, but yes. And in fact, in every case where we did this, where we took a dialkyl amino containing fluorophore and replaced it with azetidine, we saw a dramatic improvement in brightness. The exact magnitude of that change depended on the scaffold in particular, but in every case, the change was really quite substantial. I won't talk about those other scaffolds much more, because largely we were really most interested in rhodamines for some of the reasons I've talked about earlier with the open-closed equilibrium and how modular and tunable they are, which we'll get to in a minute. They're also consistently amongst the brightest of all of these fluorophores. And we wanted to know, well, okay, can we make structural modifications to take JF549, a single dye at a single wavelength, and expand that across the palette of visible light? So now I'm going to step through work that we did over a period of 7 or 8 years to modify the structure starting from JF549 to make a wide variety of what we call JF dyes. In order to do this, though, and to do it in a systematic and rational way, we needed reliable, efficient, and in some cases, new chemistry. Now, I mentioned the classic synthesis, which is not really amenable to our purposes because the azetidine doesn't survive it. But we now had the palladium catalyzed cross-coupling. We could also do a slightly different approach where we install the bottom rings of these dyes through an aryl metal, like an aryl Grignard addition to a ketone. And in 2017, we reported a new strategy where we would take these dibromides, which could undergo lithium bromide exchange and then generate this nice angry bis aerolithium reagent that would attack an electrophile twice, like an anhydride, to assemble the central ring in a single step. And this ended up being one of our most efficient ways to synthesize a variety of the Janelia Fluor dyes. Okay, so the first modification that we looked at was to replace the central oxygen atom with other atoms, like carbon and silicon and other heteroatoms like nitrogen and sulfur. And it had been established by other groups, particularly Nagano's lab, that doing this could result in pretty substantial red shifts in wavelength. And so we wanted to do this with JF549. And when we replaced the oxygen with a carbon or a silicon, that gave us the dyes JF608 and JF646. So these substitutions are buying us around a 50 to 60, and then overall about 100 nanometer red shift. We could expand that to include other heteroatoms like nitrogen, which causes a blue shift, a regular thiol, in this case a sulfide type linkage, which slightly shifts it into the red. And then these more elaborate phosphorus and sulfur moieties, which shift them far into the red. Now, these two dyes in particular are not very useful because they are extremely closed. Their KLZs are almost immeasurably low. But I'll talk in a minute about how we can rescue that and make them useful. So as organic chemists, when we look at this azetidine ring, we think, hey, well, what happens if we start sticking stuff on there? And so what we found is that by putting substituents on the azetidine ring, we could actually perturb the wavelengths of these dyes and blue shift them in nice small increments. So starting from JF549, as we put different electronegative groups on the azetidine at the 3 position, we observed a gradual and systematic decrease in the absorbance and excitation and emission wavelengths while maintaining high quantum yields. And the degree of the blue shift actually correlated with the electronegativity of these groups. But it didn't just affect the wavelength. And when we looked at the equilibrium constant for the lactone-Zwitter ion equilibrium, we saw that essentially the same type of trend, where we saw a nice decrease in KLZ, which correlated with the electronegativity of the substituents. And so that would give us dyes like JF525, which is still a relatively open dye, but it has improved cell permeability compared to JF549. It tends to get into cells substantially faster. And it's been a very useful dye for us for applications like some of the chemogenetic indicators I'll briefly mention later. Now, we could take the lessons we had learned here and apply them to basically all of these rhodamines, regardless of what the X heteroatom was, and do some interesting sort of rational design things. We could take JF608, which is the paired carbon azetidinyl rhodamine, and we could fluorinate on both azetidine rings. And what that would do is take a dye that as a halo tag ligand is not fluorogenic. It's highly absorbent and fluorescent, regardless of whether it's bound to halo tag, and convert that into a highly fluorogenic dye that's also shifted to perhaps a more convenient wavelength, just by adding these fluorine atoms. All right. So in a sort of related type of substitution, we also were interested in replacing the hydrogens on the azocyclic moieties with deuteriums. The reason for this being that we knew that one of the principal photobluing, photobleaching, decomposition pathways for rhodamines was dealkylation, and we thought that an isotope effect here could perturb or decrease the propensity for that process. And so making this type of substitution gave us dyes like JFX650. And when we compare it to the parent dye JF646, in SPT, we saw higher intensities and longer track lengths. And moreover, in more general fluorescence, like confocal microscopy, we saw higher brightness and photostability. And this is a really nice video, I think, because it illustrates this more qualitatively. So we have the azetidine, the JF646 parent dye, the deuterated azetidine, and the sort of optimal deuterated pyrrolidine. And what you see in this video, I think, here we go, is that 646 is the first to go, followed by X646. And then X650 is the last man standing. And demonstrating here that it does indeed show this improved photostability in real-life applications. All right. So one of the more recent modifications that we've explored at length is to replace the hydrogen atoms on the bottom pendant phenyl ring with fluorines, and this has several benefits. One of those is that it shifts the dye 20 to 25 nanometers into the red. So if we do this with JF646, we get JF669. But in addition to just shifting the dyes, it also causes them to shift more towards the open form. It increases KLZ substantially. So here we have JF646 and 669 free in solution, not bound to any protein or anything. And you see that 646 has very low absorbance, very low fluorescence, whereas 669, by virtue of this shift in equilibrium, now has very high absorbance. And so this modification buys us two advantages. It also comes with the added benefit that the fluorinated derivatives like 669 tend to demonstrate improved photostability compared to their desfluorinated analogs. And if you more quantitatively you look at the KLZ, you see that just installing these 4 fluorine atoms on this bottom pendant ring results in an almost 200-fold increase in the equilibrium constant. And this holds across all of the different subclasses of the azetidinyl rhodamines. And all of these different modifications are cooperative or tandem. I mean, some of them will sort of fight each other, whether you're substituting on the azetidine or putting fluorides in the bottom ring. But you can combine them in different ways to rationally design new fluorophores. And one nice illustration of that is if we go through the evolution of one particular far red dye. So if we start with JF549, we want to push this way into the red, so we put a phosphine oxide in there. But this dye has an immeasurably low KLZ. It's totally closed up. It's not really even a dye per se. Well, we know that we can fluorinate on the bottom ring to open that up, which gives us JF722. And now if we fluorinate on this ring, we can tune the KLZ back down a little bit to give us a dye, JF711, that is actually highly fluorogenic and actually also has the advantage of a slightly higher quantum yield than JF722. Okay, so just by combining all of these different structural modifications, we can actually cover a pretty decent portion of the visible spectrum. And so here I just illustrate, several or most of the good JF dyes that we've made over the years. This was sort of the state of the art, I guess, in around 2020 or so. And so we have all these dyes, some nice colors. But it's not really just about the color. As I've alluded to a little bit, several of these dyes have interesting properties. So several of them show a high degree of fluorogenicity. Others show really excellent brightness and photostability, approaching best in class. And others have really excellent permeability and bioavailability, which comes into play when you want to use these in tissue and even in whole animal. So I'm going to just really quickly run through some of the more advanced applications of these dyes. A lot of this work is several years old, so I don't want to sort of belabor the point. But with those dyes that show decent permeability and bioavailability, we were able to use them in tissue and in whole animals. And here I'm focusing on a couple of the fluorogenic dyes. So JF585 halotag ligand can actually be administered to mice through IV injection in the tail vein. These mice express GCAM6 and halotag in layer 5 of the visual cortex. This dye is able to cross the blood-brain barrier and shows co-localizes very nicely with GCAM6. JF635 halotag ligand, another highly fluorogenic JF ligand, can be used in tissue like Drosophila CNS, where here the basin neurons are expressing halotag. We administer JF635. It shows up, it's everywhere here, but we see fluorescence only in those basin neurons. We've done some more elaborate modification of the JF dyes that I'll just touch on very briefly. We can actually cage these dyes using a technique originally reported by Stefan Hell's group, where we convert them into diazoketones that we call PAJF dyes. These are stuck in the closed form, And when we expose them to UV or blue light, this falls apart and regenerates the fluorescent dye with spatiotemporal control. And several of our colleagues at Janelia have done some really cool 2-photon and super resolution experiments demonstrating the utility of these types of cage dyes. And Claire Deo, when she was in Luke's lab prior to having her own lab at EMBL, leveraged some of the JF dyes to build some nice calcium indicators that linked the calcium chelator BAPTA with the halotag ligand, so you could direct these to specific structures and sense calcium. And finally, I would be remiss, I think, if I didn't mention some of the really pioneering work that's come out of Eric Schreider's lab to develop chemigenetic indicators that incorporate the JF ligands, for example. So we have Voltron, for example, a voltage indicator. We have HaloCamp, the calcium indicator, and the more recent WhaloCamp, which takes advantage of several of the different JF ligands. That's about the extent of what I know about these things. So if you want to read more about them, check out their papers, because I'm just a chemist, so you know. So now we had a decent set of azetidinyl rhodamines, and we started to notice some interesting trends as it relates to the lactone-zwitterion equilibrium. And that was that the position of the equilibrium seemed to correlate quite well with how they would behave in biological systems. So dyes that were down here at the very lowest KLZs, those that were almost immeasurably low, are always non-fluorescent, always closed up, and really aren't very useful dyes. On the other end of the range here, high KLZs above essentially 1, you have dyes that are always fluorescent, essentially environmentally more or less insensitive and very bright. In between, we could have dyes that show improved permeability or dyes at lower KLZs that are fluorogenic. If you look specifically at the silicon analogs, what we noticed is that we could tune it down pretty much as far as we could go with JF635 to make a dye that at least is still useful. We could fluorinate on the bottom ring to make 669, which pushes us up into the improved permeability region. But this seemed to be about as far as we could go with the azetidinyl rhodamines. And so it was at this point around the beginning of the pandemic, I think, that we started to question, well, have we pushed the azetidinyl rhodamines as far as we can go? You know, what more can we do to make brighter, redder dyes using some of the lessons we've already learned? And so that required us to think a little bit outside of the azetidine box, so to speak. Of course, there are many, many different subclasses of rhodamines available to us, from the very simplest rhodamine 110 through the highly fused gelaladine-based rhodamine 101. And all of these are quite bright fluorophores. We also have the more elaborate Alexafluor and closely related Atto dyes, which have more complex nitrogen substituents, or oxychromes. And what we noticed was that in the literature, really very little had been done in terms of fluorinating the bottom ring and exploring substitution of the bottom ring on how that would perturb the properties of these dyes and potentially improve them. We were interested in the fluorination not just from the standpoint of the spectral improvements, making the dyes redder, making them brighter because of the KLZ shift, but also from a synthetic chemistry standpoint. Because these fluorines on the bottom ring could actually function sort of as latent functional handles. They're pretty robust and stable, but they undergo substitution reactions with specific nucleophiles. And so we found we could use what we call a masked acyl cyanide reagent at the end of a synthesis to displace a single fluorine in a regioselective manner, and then use this unmask it, and react it with a nucleophile to install whatever labeling motif that we wanted. And so this was a nice late-stage way to derivatize our dyes, as opposed to traditional rhodamines lacking the fluorines, where we would have to incorporate some functional handle generally at the beginning of the synthesis, which can introduce a whole host of problems with protecting groups and functional group tolerance. So our goal here was to take a variety of different rhodamine scaffolds, make the fluorinated dyes, and then use the masked acyl cyanide chemistry to make useful derivatives for labeling as probes. Of course, the question was, how do we make all of these fluorinated dyes? Because that was not necessarily obvious. And then we think the masked acyl cyanide chemistry at this point is general, but how general is it? All right, so I've talked a little bit about some of the rhodamine synthetic strategies that we had used or developed, but in order to access some of these more elaborate fluorinated ones, we had to go a little further. So one of the strategies we used was this lactol condensation. In 2016, two chemists at Promega reported an interesting reaction between these partially reduced phthalic anhydrides and aminophenols. And so we extended this to include fluorinated derivatives and silicon rhodamines to access a larger collection of rhodamines in a very simple reaction. We also found that we could take tetrafluorobenzoic acid, directly lithiate this, and install the bottom fluorinated ring by adding it to a ketone. And this solved a long-standing problem with trying to metallate tetrafluorinated aryl rings. And we very briefly dabbled in modifying the acid-mediated synthesis using, instead of protic acids, using Lewis acids to assemble some much more complicated and challenging targets, which I'll just briefly mention later. Okay, so we have these synthetic approaches. The lactol condensation was probably the most general. It allowed us to access a wide variety of oxygen fluorinated rhodamines and silicon fluorinated rhodamines. We could use this tetrafluorobenzoic acid route to access the fluorinated carbon rhodamines, which until then had been a really challenging set of targets to access. We could use our Lewis acid-mediated modification to make these highly sterically congested, bulky carbon rhodamines fluorinated on the bottom ring, for reasons I'll mention in a minute. And lastly, we still could leverage the palladium catalyzed cross-coupling to make a more limited set of rhodamine 110 analogs and some N-aryl rhodamines, which can function as quenchers. And I don't want to belabor the point here, but the fluorination is very consistent in its impacts on the chemical and spectral properties. So when you go from hydrogen to fluorine on these dyes, you always see a red shift. You always see an increase in the KLZ towards the Zwitter ion. You see an increase in the extinction coefficient and a modest decrease in quantum yield, which is largely to be expected based on the energy gap law. And just to sort of exemplify this, if we compare these two fluorinated dyes to their non-fluorinated congeners, we see that indeed the wavelengths are shifted by around the same 20 to 25 nanometers. The extinction coefficients are dramatically higher, and the KLZs improve by 20 to 100 fold just by fluorinating that ring. And not surprisingly, the masked acyl cyanide chemistry worked fine with everything we threw at it. And so we were able to convert all of these dyes into halotag ligands, and many of them into snap tag ligands and CP snap tag ligands. We even made hoaxed derivatives for binding to DNA that I just don't show here because they're huge and take up too much space. But this basically worked in every single case that we tried. So it's a really nice way to introduce these handles at the very end. So at this point, we had a pretty comprehensive collection of dyes if you took our work prior to this most recent set of dyes in combination with the new ones. And so at this point, we wanted to, again, revisit our sort of general rubric here and look at how the equilibrium constant affects their position on this scale, or their behavior, I should say. So as I mentioned before, we have 646 and 669, where the fluorination shifts us up into this region here. But when we switch to a different oxychrome, the silicue rhodamine, that has a slightly higher KLZ, and fluorinating it pushes it way up into the very bright, always fluorescent region. And we see the same trend with the gelaladine-based rhodamine 101s. And JF698, which is the fluorinated analog here, is one of the most open silicon rhodamines that we've made. It might be the most. And it compares very favorably to some of the brightest 700 nanometer dyes around. Okay, but that's just the silicon examples. And what we found when we looked at the data more broadly is that this trend was very consistent. So of course the fluorination shifts the KLZ, we know that. But as we move down this list of oxychromes, or nitrogen substituents here, we found that the KLZ ranges would shift more towards higher values as we move to more electron-rich oxychromes. And so that allowed us to cover more ground even with the same, say, silicon rhodamine scaffold. This was true for the carbon rhodamines as well, and even for the oxygen ones, although here you start to run into the limits of what we can measure with the KLZ because they all become quite compressed. They're all very open, and yeah, we see the same general trends, but it's not quite as pronounced. All right, so I know I'm running over, I apologize, but I just have a couple quick examples to illustrate how these lessons are useful. So I'm going to start with one, focusing just on how the fluorination and the oxychrome modification can just make bright, new, far red fluorophores. So if you start with the most extreme example, which is rhodamine 110, silorhodamine 110, this is a yellow-orange dye with a very low extinction coefficient and low KLZ. And when we incorporate the most electron-rich oxychrome and fluorinate the bottom ring, here we've dramatically shifted the dye all the way out to 700. We have a much higher extinction coefficient, largely as a consequence of the fact that the KLZ is now almost 5, which is quite large for a rhodamine. And when we went ahead and looked at the properties of some of these new far-red, near-infrared JF dyes, we saw that they showed substantially improved brightness and photostability compared to some of our older spectrally similar JFs. So with JF698 halo tag ligand, when we compared it to several of the others, we saw it had 3 times higher fluorescence or nuclear intensity, and substantially higher photostability compared to even another dye from this work, as well as the phosphine oxide JF dyes from our earlier work. And Ariana and T3 was able to do some really nice 4-color imaging, taking advantage of the spectral separation here and incorporating 3 different JFX dyes, or JF dyes, from 549, 650, and 698. So the fluorination is not just useful from the spectral property standpoint, but as I mentioned before, from a synthetic chemistry standpoint. And it can be used to fix dyes that have particular issues. And one of those dyes is ATO647N. So ATO647N is one of the very best dyes for single molecule imaging, but kind of sucks for live cell imaging. And that is because of how it is conjugated for attachment to whatever biological structure you want to study, where its amidation of this carboxyl group here locks the dye in the open form, leaving it as a cationic species. And that causes the dye to localize to the mitochondria. So despite the fact that halo tag is expressed here inside the histones, you only really see the ATO647N in the mitochondria. Well, we thought, well, what if we fluorinate this dye, which is otherwise a really great dye, and use the mast acyl cyanide chemistry to install the halo tag at the more preferred and standard position at the 6th position of the bottom ring. And this fixes this issue because now we still have a zwitterionic net neutral dye, and this is actually the more optimal position for the enzyme itself. When we did the same experiment, JF657 neatly co-localizes with Hoechst in the nucleus, meaning it's binding to halo tag on the histones. James Liu's group has also started to look at it as a single particle tracking dye. And when it's compared to sort of our previous state-of-the-art dye, JFX650, JF657 shows a lot of promise, where it's actually brighter than JFX650 and shows more consistent localizations over time, indicating perhaps slower bleaching, although we suspect there may also be some blinking activity at play here. So this is my last little nugget about how we can use this type of chemistry to fix dyes. So N-aryl rhodamines have been known for a while to be potentially useful non-fluorescent fret quenchers or acceptors, principally due to their very low KLZ, which results from twisted internal charge transfer with those aryl groups. But this N-arylation tends to firmly trap these dyes essentially in the lactone form. They have KLZ values that are vanishingly low. But we have the fluorination, of course, and so when we fluorinate that dye to generate what we call JQ645, now we have a much more reasonable KLZ. Reasonable enough that when we make the halo tag of this dye, we have a highly fluorogenic ligand that is 50-fold fluorogenic upon binding to halo tag, and also labels halo tag much faster than the non-fluorinated derivative. So T3, particularly Tim and Ronick, were able to make use of this dye and construct a semi-synthetic cyclic AMP indicator, which we call SCAMPy. This works by connecting snap tag and halo tag through EPAC1, which is a protein that binds to cyclic AMP. We label snap tag with a bright dye like JFX612. We label the halo tag with the quencher. When in the absence of cyclic AMP, these two regions are close together, and so there is substantial fret, and the fluorescence lifetime of the dye decreases, or the fluorophore decreases substantially. Now, when cyclic AMP concentration increases, or when it binds, this moves these regions apart, decreasing fret and increasing the fluorescence lifetime of the donor fluorophore. And you can see that pretty nicely here, where we see this statistically quite significant increase in fluorescence lifetime. All right. So that's about it. To keep this friendly for YouTube and public viewing, I'm not going to talk about our future work right now. But I do want to, of course, acknowledge a bunch of people. First and foremost, Luke, of course, for all of his guidance, mentorship, and support over the last 14 years, believe it or not, that I've been in his lab. I especially want to thank the open chemistry team. Carla and Sarah are indispensable in maintaining our dye distribution program that I think most Janelians are probably familiar with. They resynthesize, QC, aliquot, and package the dyes for shipment. And Anastasia has the unenviable task of coordinating all of these shipments. We're up to about 3,000 plus orders at this point. So that is a lot of work. Anastasia also makes our lives so much easier through all the logistical work that she handles that makes our lives so much easier. I want to thank the rest of the Lavis lab for being a great group of scholarly, considerate coworkers. Brian has contributed tremendously to a lot of the work that I've shown here today, as has the entire tool translation team. And without them, all we'd be doing is making dyes that would just sit in a freezer and nobody would use because no one would know what they do. I also want to thank several alumni that have come through the lab. We've had a really successful post-bac program over the last 10 to 12 years. And that has included Anand Muthuswamy, Joel Slaughter, Anthony Ayala, Natalie Falco, and most recently Katie Holland, who have all contributed to the work I've shown today and been just indispensable in certainly making my life and Luke's life a lot easier. And I'm not going to even try to list all of our collaborators because we have so many excellent ones here at HHMI at large and externally. Yeah, you guys know who you are, and we really appreciate your work. So that is about it. I am sorry I ran a little over there. Hopefully, maybe people got a good nap. But yeah, I didn't put a slide in here to specifically mention the open chemistry and dye distribution program. But if you are interested in trying any of these dyes, particularly some of the new ones, which we're just now starting to add to the website, you can check those out at dyes.janelia.org. You can also contact us by email or through whichever social media hellscape you prefer. And that's about it. Yeah. If there's any questions? Anything? Anything?
I have a question, if I may.
Yeah, sure.
Okay, yeah, thanks, John, for this excellent presentation. Really impressive work, and thanks also for the quite educative presentation. And so it's great to see how you can use these modifications to tune the spectra and other properties, right, of these dyes. And I was also happy to see that some of these are really well suited for in vivo delivery, right? But maybe I missed it. I was wondering, do you know which type of modifications actually improve the bioavailability and make this in vivo labeling more efficient and more successful?
So I think that partly we have a rough guide to how we can do that based on the position of the open-closed equilibrium for these dyes. So if we measure the KLZs, we can kind of guess which of these dyes might be more amenable to having higher bioavailability. There is a certain degree of, I think, idiosyncratic or empiricalness to it, but we can tell generally just based on where that equilibrium sits as to how bioavailable they might be. So the dyes that are reasonably fluorogenic or sit within that sort of enhanced cell permeability region, those tend to be some of the better ones as far as bioavailability is concerned.
And sort of, do you think there's further room for improvement of making more of these dyes that are bioavailable?
Oh yeah, I mean there's there's an enormous number of dyes we could make with these modifications, and the number of combinations of the different oxychromes and substituents, fluorination, deuteration. There's tons that we could do to make more bioavailable dyes. It's just sometimes an issue of time and manpower, I would say. But sometimes there are even surprises, like JF669 is a dye that I think when we first made it, we didn't necessarily inspect that it would be as good as it is, but it is a terrific dye that is highly bioavailable and permeable. So some of it is, a little empirical, but there's just a lot to do and many combinations that we can continue to pursue. I don't know if that answers your question. But.
Oh, thanks, and keep up the really great work. It's really exciting.
Thanks.
I see also a question in the chat. So Olivia is asking, or is commenting, thanks for the great talk and the open chemistry. Sorry if I missed it, but did you measure the properties of Janelia fluor dyes in cryogenic conditions? Is the performance the same?
That is not something that we've really looked at, right? Yeah, I think there was one paper from Harold where we might have looked at it a little bit, but that is a bit of uncharted territory with the JF dyes. We do have some ideas in the pipeline for things we might do to look into that, but as of yet, that's kind of more of a frontier for this work.
Yeah, thank you.
So John, great talk. I have a question that I realize is sort of outside your preview, but it's in the field of chemistry, which is in vivo chemistry and the toxicity effect.
Mm-hmm.
So I could see all sorts of potential applications in clinical use of some of these dyes, not the halo tags probably. But do you have any sense of toxicity of the different modifications?
I mean, I don't think rhodamine dyes are great as far as toxicity is concerned. When you talk about some of these structures that have a bunch of anilines on them, they tend to have some adverse effects. I certainly wouldn't want to inject myself with a bunch of them. So I think
So you get injected with fluorescine, right? So that's very safe. And most of the things that we've injected in mice haven't seen any issues. One of the biggest issues with these injections, these things are usually not soluble in water in high concentrations. So we're actually worried about DMSO and stuff. And we looked at whatever we can look at in mice that people usually look at toxicity and haven't seen any effects. But it could be that, we probably looked at the safer ones first and not the really crazy ones.
So I'm just focused on the IR dyes, and I've forgotten the name of it, but it's a well-known IR dye, clinically approved, always used for putting it into the blood, and it's got a really terrible quantum yield, but clinically approved, so everybody uses it, and so there's a lot of opportunity. This is not a business, but it just seems like there's opportunity for imaging, hematomas or something like that in vivo, in clinical application. And so that's why I just asked. So.
Yeah, no, I mean, I think there is certainly potential there. I mean, I think we have to take a longer look at toxicity, metabolism, pharmacokinetics, to really know for certain whether they have...
You're the man.
whether they have a potential there. But no, it's definitely something to think about.
Yeah, that's a tricky field. I know. But collaborators are the ones that, so...
Yeah. I think the issue is the dye you're talking about is enzyme green, ICG. Right?
Yeah, ICG, that's it.
Yeah. And, you know, the problem is the FDA approved that in the 50s. And so then
Wouldn't approve it
everyone's
today.
like, yeah, they would never approve it today. So then everyone's like, how can we use ICG? And, you know, and so everyone's just doing that, right? And so it's, I think, I don't know if there's been a new sort of tracer dye that's used in humans approved
I don't think
in the
so.
last 7 decades. I don't know. You know, we have a good collaborator and friend, Martin Schneerman, up at NCI, who's closer to this and thinking about, you know, fluorescence assisted surgery and other things. And it's, you know, it's challenging, right? So we have sort of shied away from that, especially because most of the dyes are cyanine dyes that are used in humans, and we don't work on those as much.
Yeah, I mean, I would be very curious, especially as we've made some of these greasier aniline-based rhodamines. I think things like HERG would be a big problem, IKR. And since industry and the FDA is very concerned about that could be a big problem. So, at least with the rhodamines.
Let's thank John again. It was amazing. Thanks so much, John. And come back February 27th.